The topic of this presentation is basically related to the pathogenesis of adenomyosis, and it has to do also with its implication in infertility. I totally agree with you. The subject is hot, and it's hot in many ways. For me, there are two main reasons. First, the image of adenomyosis is blurred by many contradictory data. Secondly, the infertility itself, well, it's the place where there are so many questions unresolved, and basically they do have, let's say, ties with endometrium, particularly with the endometrium. Although the landmarks of the definition were set almost, 50 years ago, the definition is still problematic. And even among pathologists, as you can see, they do not agree on what the glandular extinction means to put the diagnosis. Only recently, and well, we know very well this, only recently imagistic techniques have replaced the invasive procedures. And in this respect, we owe a lot of respect to Dr. Exacustos and Dr. Luciano for their extended work that today allows us to put a diagnosis of adenomyosis without using invasive procedures. But still, these investigations are quite expensive, and I also have to say that they do require an experienced practitioner. So we have all the reason to suspect that the diagnosis of adenomyosis, well, we have all the reasons to believe that it's underdiagnosed. The association between adenomyosis and infertility has been reviewed in several systematic studies, and as you can see, the results were pretty similar. Actually, to conclude, we might say that the association is possible, it's probable, but it's not certain. So let's review the current data. We do not have any study to analyze the impact of adenomyosis on natural conception or in IUI procedures. We do, on the other hand, have quite good evidence regarding the impact of adenomyosis in IVF procedures, and we also have data regarding the impact of adenomyosis in patients with endometriosis. Now, regarding the IVF procedures, first meta-analysis couldn't reach to significantly results, basically the lack of data. However, Vercellini managed to obtain good data, good enough to assert that adenomyosis decreased the risk of pregnancy, as we all know, and at the same time increased the risk of abortion. However, I want to note that when reducing patients with more than one procedure, those results lack statistical significance. So I want to pause here to make a point to what I consider to be a very important thing in this enterprise. When analyzing this IVF procedure, considering the type of protocol used, we can observe that actually in the antagonist protocol, all the studies recorded a detrimental effect of adenomyosis in pregnancy. On the other hand, when considering the long protocol, there are no more significant differences between controls and adenomyosis. Even more, when we analyze when we get to the donor programs, we can see that actually in those patients without a pre-treatment with agonist, the rate of abortion was actually higher than in normal population. This problem was fixed the very moment we added, actually, there are two authors that had this, they they did these studies. When adding agonists, there are no more differences in terms of abortion comparing with general population. And at last, Vercellini did a wonderful study on patients with colorectal endometriosis, and he obtained very strong data. Actually, the impact of adenomyosis in infertility was very strong. So, to conclude, we may say that at least some patients with adenomyosis do encounter infertility problems, and at the same time, that adenomyosis, at least part of the problems related to adenomyosis, could be fixed by giving agonists. Regarding the pathogenesis, basically, every system that has to do with the control of the endometrium could be a possible culprit in this process. And in this respect, I intend to go through the steroid metabolism, junctional zone, interleukins, integrins, free radical oxide system. Now, although it looks very much, when eliminating studies without statistical significance, we obtain quite a small number of cases, somewhere 50, 60 around. And first, I will start with the junctional zone. We identified primary changes in this territory, changes that are related to the transmission of the information, and at the same time with the oxytocin receptor. We also observe an unnatural reaction to the estradiol. And what it is also interesting, medical treatment not only corrected the symptomatology, but also reduced the thickness of this territory. Another issue that I think is very important is related to the impact of this territory in implantation. Independent of adenomyosis, the presence of a thick junctional zone was systematically associated with a decrease in the IVF procedures. Regarding the metabolism of endometrium, we analyze the production and also the receptivity. And in terms of production, most of the studies were focused on aromatasis. We have first studies proved obvious an increase in aromatasis, but pay attention, all those cases required hysterectomy. So it means that probably it was a severe form. At the same time, we did indeed observe increased aromatase, but it was also not very specific. Other estrogen diseases, pathologies, also showed the same pattern. And there are another two issues, I think, very important. There was a direct involvement of these changes in the production of aromatosis in the rate of pregnancies. And also, they were strongly correlated with the extent of the symptoms. Now, we do have only 2 studies regarding the sulfatase. They were also increased, but not specific for adenomyosis. And we have identified something like 5 studies regarding mutations in the receptor, whether it was estrogen or progesterone. Again, the same situation, not specific for adenomyosis. With respect to immune system, we have identified only 2 studies regarding the expression of human leukocyte antigen. And at the same time, they were not specific, and they were identified in severe forms of adenomyosis. And also very important to notice that actually when LHG was increased, there were strong chance to have repeated implantation failure. Regarding the cells, we have only a few studies. Only a few studies when, of course, we reduce those without statistical significance. And these studies were very much alike. So you can see that actually uterine natural killer were increased in adenomyosis, but only in severe forms, whereas mild forms and controls, of course, were normal. At the same time, we identified an imbalance between a lymphocyte, regular and helper, and the same story. They were basically related to the intensity of dysmenorrhea, and the macrophages were also increased only in severe forms. Now, about interleukins, we have identified many studies. What is interesting, well, they were all changed. However, 8 out of 11 studies, we can assume that there were severe forms as hysterectomy was required. And on the other hand, for the other 3, the control group was fertile patients, where actually the group of study were infertile adenomyotic patients. With respect to integrins, we have only 2 studies, and these 2 studies show the same time changes in this production, but not specific for adenomyosis. And at last, concerning the free radical oxidation system, most of the studies were conducted by OTA. All the results were pretty much alike, so actually he identified changes, important changes in patients with adenomyosis. But again, same story. All these patients required hysterectomy. When we analyzed patients which, the diagnoses were made by a non-invasive method, there are no more differences. So when we draw the line, we basically notice a pattern that is repeated virtually in all the system. So in early forms of adenomyosis, we couldn't find any recurrent abnormalities. Whereas in severe forms, all the parameters were changed. And another thing that I think is very important, the clinical symptoms were not related to adenomyosis or only a mild way, but primarily were associated with the primary dysfunction, such as rheumatism, the dysfunction we discussed before. So actually, these results, we were looking for a parameter that might help us to describe adenomyosis, and obviously we failed, which is disappointing, but actually it's by no means unexpected. We know very well that when analyzing the functionality of the endometrium, standard procedures failed to produce consistent data. And on the other hand, omics, and particularly transcriptomics, seems to be indeed a research field full of promises, basically due to their capacity to analyze more data at the same time. So in this respect, we do have 2 small-scale studies on adenomyosis. Both of them were conducted on patients who required hysterectomies. And the results were pretty much similar. Actually, there were hundreds of genes up and down regulated, genes which are related to the structures we discussed before. So the authors speculated that perhaps some of these alterations were, let's say, the result of a sort of a domino-like effect. And to substantiate this point of view, we turn to studies on Hox genes, which we all know that are essential for implantation. And altered Hox genes were basically identified in all patients, infertile patients, endometriosis, leiomyoma, polyps. Correcting the underlying problem prompted a change, an improvement, an immediate improvement in the expression of these genes. To summarize, current data, according to our opinion, calls for a change in the perspective of what adenomyosis is. Traditionally, the pathogenesis of adenomyosis was regarded as a linear process, which started with a discrete dysfunction, continues with an important dysfunction, and eventually it leads to clinical symptoms. Now, the new scenario, which is, pay attention, a nonlinear process. And after all, this is by no means surprising. Biology is a nonlinear process in most of the cases. So in this new scenario, there is a mutation. Everything originates with a mutation, punctual mutation. And this will lead, perhaps could lead to symptoms, but at the same time, it opens the gate for adenomyosis. Now, the occurrence of adenomyotic foci is not necessarily, it's not automatically related to the alteration of the other systems or the aggravation of the symptoms. Nevertheless, when a critical mass is reached, from that moment onward, it will exacerbate a primary dysfunction. It will inflict all the other systems and control the endometrium, and in the end, it will provoke massive symptoms. So to conclude, adenomyosis is a highly heterogeneous disease. There are strong evidence to offer a special place for the junctional zone in this whole enterprise. And given the wide array of pathological possibilities, it's only too logical to try to narrow the area of investigation in order to tailor the treatment. And in this respect, I will propose to you actually the only classification that was tested from a clinical point of view. And as you can see, it was proposed by Kishi. And although in symptoms, they look very much alike, you can see that the behavior is very different. And accordingly, the treatment was different. Thank you.